Hello everyone, I'm Rayleigh.
And I'm Monty Sakarvadia, and today we're going to be presenting the results of our study, which is shown on this poster. And we will be presenting this poster in the form of a short slide deck. So our study was titled Atypical Neonate Extra-axial Cerebrospinal Fluid is associated with reduced cognitive development at age 1 and 2 years. So the objective of our study was to assess if atypically enlarged EA-CSF volumes at neonatal age is associated with a child's performance on the Mullen scores of early learning assessment at 12 years or 12 months and 24 months of age. We formulated this research question based on a paper that came out of our lab by Shen et al., published in 2017, which discussed that atypically enlarged EA-CSF was associated with autism spectrum disorder diagnoses of a child later in life. So our hypothesis focused on the idea that there will not be any major associations for typically developing kids between their EA-CSF volumes and their Mullen scores of early learning assessment outcomes. However, for atypically enlarged EA-CSF, we will be seeing reduced cognition based on lower scores on the Mullen scores of early learning assessment. So methods and materials for the study consisted of 3T MRI scans, which we acquired from 651 infants, 300 females, 351 males at neonate age, approximately 3 weeks old after being born. And the pregnant women, a part of this study, were all recruited from UNC hospitals and Duke University Medical Center. The Mullen scores of early learning assessment was administered by a professional to these children at 12 months and 24 months of age. And the criteria that this assessment measures is gross motoric, fine motoric, expressive language, receptive language, and visual reception. And all of these categories are then combined into a composite score except for gross motoric skill. And to analyze our volumes and how they relate to the outcomes of the MSEL, we use general linear models. And this figure in the bottom right just shows what our MRIs looked like when we were initially extracting the EA-CSF volumes, which is red at infancy.
Here on the left, it's a histogram shows us the distribution of the EA-CSF volume, and we normalize the volume by dividing the intracranial volume, which make the variable independent of the head size. Also, the two dashed lines here shows the top 5% and top 10% of the distributions. And on our preliminary analysis, we tried to find the relationship between the normalized EASS volume and the Mullen score. However, we can see that there's no clear relationship for the whole populations. And that's why we focus our further analysis on the extreme value. And again, here the two dashed lines indicates the top 10% and top 5% normalized EASS volume.
So our results for our analysis with generalized linear models are here on this slide. In our general linearized models, we were trying to predict Mullen scores of early learning based on EA-CSF volumes. And the covariates we used to do this were intracranial cavity volume, gestational age at birth, mother's education, age at MRI, age at the Mullen scores of early learning assessment, absolute time of MRI, and sex. How we did this analysis is we first split up the whole cohort into the bottom 95th percentile and top 5th percentile with respect to their EA-CSF volume at infancy. And then again, we did the same thing, but with top 10th and bottom 90th percentiles. So for the top 5th percentile at 1 year of age, we were finding that there was a significant negative association between their EA-CSF volumes and the expressive language and cognitive composite scores on the MSEL. And then again, at 2 years of age, there was a negative association between that volume and the receptive language. And all of the results from our GLM can be seen in the table right underneath. And then for the top 10th percentile, we found that at 1 year, there was a negative association between EA-CSF volumes and then fine motoric skills and cognitive composite score on the MSEL. However, at 24 months of age, we didn't find any significant negative associations. But again, you can see our values on this table underneath.
Here, it shows the results of linear regressions model, and it gives us some visual review of the data. On the left, we can see there's a negative relationship between the Mullen score at first years and top 5% of normalized EA-CSF volume. And on the right side, after we remove the outliers on the right corner, there is a negative relationship between the expressive language and 2 years and the top 10% normalized EA-CSF volume.
So to discuss and conclude our study, we found that there's a missing association between typically developing kids and their outcome on the MSEL, which confirms the findings from Shen et al. EA-CSF at infancy does not appear to be a biomarker characterizing typical brain development. However, atypical EA-CSF enlargement seems to be indicating atypical brain development, or at least an increased risk for atypical brain development. We suspect there may be a pathological threshold of high EA-CSF volume, or at least what might be considered high. Currently, we've only investigated top 5th and top 10th percentile, but moving forward, we would love to see if there's another threshold that is more significant. And potentially, EA-CSF might serve as an early biomarker for reduced cognitive ability at 12 and 24 months. Some limitations to our study included low sample size, which might mean that in the future, we hope to focus on high-risk recruitment for children who are at higher risk for cognitive disorders and disabilities. Additionally, our analysis focused on only a single EA-CSF global volume measurement. However, if you refer to figure 4, you can see that EA-CSF volumes are not distributed evenly around the brain. Some regions may be more dense and more concentrated than others. So moving forward, we would like to do a more in-depth local analysis of EA-CSF and see if any local regions of the brain with respect to EA-CSF are more correlated with the outcomes on Mullen scores of early learning assessment. Furthermore, we want to follow up on verbal versus non-verbal quotients on the Mullen scores of early learning and see if EA-CSF is playing a role in that. Furthermore, we want to follow up with these same children in the study and see if they were later diagnosed with other cognitive disabilities, disorders, or health problems. And furthermore, we want to do a longitudinal study and see if these volume measurements at infancy might normalize over time or might not normalize over time, and how that normalization process might affect brain development and cognitive ability for that child. Thank you for listening to our results from our study, and we hope you enjoyed and
Thank you.